Hey Guru Nation, you asked for it and I'm here. I did some research just to make sure I got the actual definitions for you. What is the difference between a phase 1A and a phase 1B? And then same thing for a phase 2A and 2B. So Carla Vera Navas, who actually teaches, she's a phase 1 expert. She teaches on our CRA Academy as a guest lecturer. I'm about to do another podcast with her. Shout out to her. She did a whole lesson on this. So in a phase 1A, it's a single ascending dose study. Small groups of patients are given a single dose of the drug while they are observed. Usually it's about 3 patients per cohort at a single dose. If they do not exhibit any adverse side effects, the pharmacokinetic data are roughly in line with predicted safe values. The dose is escalated, right? So a new group of subjects is given a higher dose. If unacceptable toxicity is observed in any of the next patients, an additional number of patients, usually another 3, are treated at the same dose. This is continued until pre-calculated pharmacokinetic safety levels are reached or intolerable side effects start showing up, at which point the drug is said to have reached the maximum tolerated dose. If additional unacceptable toxicity is observed, then the dose escalation is terminated, and that dose, or perhaps the previous dose, is declared to be a maximal tolerated dose. In phase 1B, it's multiple ascending dose. So multiple ascending dose studies investigate the pharmacokinetics and pharmacodynamics of multiple doses of the drug, looking at safety and tolerability. In these studies, a group of patients receives multiple low doses of the drug, while samples are collected at various times. The dose is subsequently escalated for further groups up to a predetermined level. So that's multiple ascending dose as opposed to single ascending dose. Now in phase 2A studies are usually pilot studies designed to demonstrate clinical efficacy or biological activity proof of concept, whereas phase 2B studies determine the optimal dose at which the drug shows biological activity with minimal side effects. So I went and did some more research online. Somebody from ParExcel in a message board commented, phase 2 is patient test and dose finding. So from the phase 1, you know that your compound is effective and safe to use, but you don't know the dose that it's effective. Phase 2A is the first trial in patients with several doses, and phase 2B, they narrow the dose down to a more final dose. Then you need 2 confirmation trials of that dose to get the compound registered on the market, which is phase 3 and all that stuff. So it's a little bit tricky when you get like phase 1 and 2. I understand in 2A, they're trying to still find this effective dose. Phase 1A and 1B is about finding the safe dose, or at least a range. Phase 2A is about finding an effective dose. And then phase 2B, they already found that effective dose, so they're just doing it at that dose. In my opinion, phase 1B, multiple ascending dose study, is the trickiest to understand because it's basically the same thing as the single ascending dose, right? It's just the same cohort. It's multiple ascending dose for the same cohort, as opposed to the previous cohort on the single ascending dose. So again, single ascending dose, small subjects given a dose usually in cohorts of 3. The next group is given a higher dose. Multiple ascending dose, which is phase 1B. In these studies, a group of patients receives multiple low doses of the drugs, and they're collected at various time points. So they're testing out the different doses, multiple low doses, to see what the PK is like for the drug. So phase 1A, same dose, single ascending dose, just different cohorts. Phase 1B, the same patient could be receiving multiple levels. That's, to me, the trickiest part. Hopefully this helps. Let me know what you think. I really need to interview Carla Vera Navas again, because she goes for an hour and a half on this topic. Good luck, Guru Nation. Hopefully you're less confused now. Take care.